Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, inside every single one of your cells, right now, there's a kind of molecular factory running around the clock. It's an incredible operation that takes the abstract blueprint of your DNA and translates it into the actual physical machinery that keeps you alive. This process, well, it's called translation, and it's how every protein in your body gets built.
And that really is the central question, isn't it? It's at the very heart of molecular biology. Your cells hold this enormous library of genetic information, first stored in DNA and then transcribed into a portable message we call messenger RNA. But how does that static line of code actually create the complex, dynamic, three-dimensional machines that do, well, everything in your body? Let's dive in and take a look.
So, to really get a grip on this, we're going to follow the entire journey from start to finish, and we'll break it down into 5 key stages. We'll begin with the blueprint itself. Then we'll see how the raw materials, the building blocks, are prepared. After that, we'll watch the factory itself get assembled, see the protein chain being built step by step, and finally, witness the finished product as it's released.
Okay, first things first. Let's get a clear handle on what we're talking about when we say the blueprint.
So, translation takes place out in the main body of the cell in what's called the cytoplasm. And here, a molecular machine, the ribosome, grabs onto that mRNA blueprint. Now, just like reading a sentence, the ribosome has to read the code in the correct direction. In this case, it's always from one end, called the 5' end, to the other, the 3' end. And as it reads, it builds the protein chain with that same rigid directionality. This rule, it's really the fundamental grammar of how to build a protein.
The whole thing unfolds in 5 distinct phases, one leading directly into the next. It all starts with activating the raw materials. Then comes initiation, where the assembly line is set up. After that is elongation, the actual building phase, followed by termination when the process stops, and finally there's often some post-production tweaking.
Before any construction can even start, the building blocks, the amino acids, have to be correctly prepared. They need to be assigned to their specific delivery vehicles. This crucial first step is the activation phase.
This preparation is a 2-step process. First, an amino acid gets a jolt of energy from a molecule you might have heard of, ATP, which is the cell's universal power source. Then, this newly energized amino acid gets attached to its specific delivery truck, a molecule called transfer RNA, or tRNA. This whole process is called charging the tRNA, and it makes sure that the right building block is loaded onto exactly the right truck.
Now here's what's so incredibly important about this step. This charging reaction is the most critical quality control checkpoint in the entire system. Why? Because if the wrong amino acid gets attached to a tRNA, the ribosome itself has no way of telling. It just trusts that the delivery is correct. So the integrity of the whole process, the fidelity of the translation, it all hinges on the precision of this one initial step.
Okay, so our building blocks are prepped and verified. Now, the cell has to assemble the entire production machinery right on top of the mRNA blueprint. This is the initiation phase. And to get this factory up and running, we've got a few key players that need to show up. Of course, we have the mRNA. That's our blueprint. Then there are the 2 ribosomal subunits, the 40S and 60S, which are the 2 main parts of the factory itself. We have initiation factors, which act as the assembly crew, putting it all together. There's a special initiator tRNA, which brings in the very first building block. And finally, we need GTP to provide the energy to kick the whole operation off.
So the assembly process starts by forming what's called a pre-initiation complex. You can see here how the small 40S part of the ribosome links up with the assembly crew, those initiation factors, and that first specialized tRNA. This whole package is now geared up and ready to find the official starting line on our mRNA blueprint.
This complex then latches onto the mRNA and starts scanning along it, looking for a very specific 3-letter sequence, AUG, which basically screams, "Start here." As soon as it finds it, the large 60S subunit is recruited and clamps down on top. This locks everything perfectly into place, forming the complete, functional ribosome. The factory is now officially open for business.
And now we enter the core of the operation, the main event. This is where the ribosome will start moving along the mRNA, reading the code, and adding amino acids one by one in a repetitive, cyclical process we call elongation. So the cycle kicks off when a newly charged tRNA arrives. Think of the mRNA's three-letter code, the codon, as a sort of lock, while the incoming tRNA has the matching key, called an anticodon. This key has to fit perfectly into the ribosome's acceptor slot, or A site. It's a beautiful check that ensures the right amino acid is being delivered every single time.
And here, this is the magic moment. The ribosome itself acts like a tiny welder, forging a strong chemical link called a peptide bond. The growing protein chain, which was waiting in the neighboring P site, is snipped from its old tRNA and attached to the brand new amino acid sitting in the A site. And just like that, the protein chain gets 1 unit longer. To complete one full cycle, the entire ribosome then shunts forward exactly 1 codon, just 3 letters, down the mRNA. This physical movement bumps the tRNAs over 1 slot each, and, crucially, it clears out the A site, opening it up for the next delivery. The assembly line is now reset and ready for the next round.
This cycle of elongation doesn't just go on forever. It continues, codon by codon, until the ribosome hits a very specific signal telling it to stop. So, now we arrive at the final phase, termination of the synthesis and the release of our brand new protein. So what happens is, the ribosome eventually hits one of 3 specific stop codons, sequences like UAA or UAG. For these codons, there's no matching tRNA that can fit. Instead, a specialized protein called a release factor slots into the A site. It's like trying to fit the wrong puzzle piece in. It doesn't quite work, and it jams the whole machine to a halt.
The arrival of this release factor is the signal that triggers one last chemical reaction. It uses a water molecule to precisely snip the bond that's holding the finished protein chain to its very last tRNA carrier. You can think of this hydrolysis as a molecular pair of scissors, cutting the thread and finally freeing the new protein from the factory.
But the protein's journey isn't quite over yet. Many of them contain something called a signal peptide. It's a short sequence of amino acids that basically acts like a postal code. This code is recognized by other molecules in the cell, which then make sure the new protein gets transported to its correct final destination, whether that's the cell membrane, another organelle, or even outside the cell entirely.
And with the job done, the entire factory disassembles. The 2 ribosomal subunits separate from each other, the mRNA blueprint is released, and all those tRNA delivery trucks go their separate ways. It's just a perfect example of the cell's incredible efficiency. Absolutely nothing is wasted. All of these components are now free and ready to be recycled for the next round of protein synthesis.
So, we've just walked through a molecular process of truly breathtaking complexity and precision. One that's happening constantly, trillions of times per second throughout our bodies. Which leaves us with a final, profound thought. Given the sheer scale of this operation, what are the consequences for an organism when a single error, just one misplaced letter, appears in that genetic blueprint?